reminder of things you already know. For some people, this might be new information. But as Lisa said, the genetics is why we're all here. So it's kind of good just to all start on the same page. There we go. So first, I have no conflicts of interest to disclose, and I just want to note that this information should not be considered medical advice. All right, so a quick outline. First, we'll do an introduction to genes and to CACNA1A in particular. Then I'll go through the different types of variants that we can see and a little bit about what a variant is. We'll talk about inheritance patterns in CACNA1A. And finally, some of the effects of these variants. So first, our introduction here. So our bodies are all composed of DNA. DNA is the instructions for making all the different components of our bodies, specifically proteins. DNA is just a long string of letters that then codes for all of these things like blueprints or recipes for these proteins. DNA gets packaged into these things called chromosomes, which you can kind of see on the top left of the slide. And then a gene is just one section of DNA that codes for a particular protein. So if DNA is like the full set of blueprints or recipes in the body, a gene is a single recipe or a single gene or a single blueprint. And then you can see in that colored part, the DNA is composed of 4 different letters, A, C, T, and G. And what happens in the body is that it gets converted into something called mRNA. And then the body takes 3 at a time, 3 of these letters at a time, and converts it into amino acids, which are the building blocks for creating the actual protein. So each of these correspond to 3 letters in the DNA and the mRNA. And then once all of these amino acids or building blocks are strung together, they create the fully folded protein. In this case for CACNA1A, it's a channel that allows calcium ions to come through. It can be open or closed. So it has a particular function once it's folded correctly. So what does the CACNA1A gene do itself? So again, it codes for part of a calcium channel. And this channel allows calcium ions to enter a cell. Kind of works like a gate to say when those ions can come in and when they can't. And in turn, it causes neurotransmitters or different communications to be released from one neuron or brain cell to the next. So kind of an analogy is that it's like a traffic cop in the brain for when these messages or signals should be sent from one neuron to the next one. So I have kind of a picture of what this looks like. If we zoom in really close in the brain and really close in between those 2 neurons or brain cells, we have this, the end of one brain cell to the next one. And they have those circles that you can see, those vesicles that have the neurotransmitter. That's sort of the signal or message that gets sent to the next cell. And what the calcium does is when it allows, or what the calcium channel does, is that when it allows calcium to enter, that message can then be released and received by the next neuron. And so it can allow those communications to be passed from one cell to the next. In a little bit simpler format, if we look at our brain and we have our string of neurons all next to each other, they might have like a communication or message that's ready to be sent at the right time. And our calcium channel, or CACNA1A, kind of acts like this traffic cop to say, when it releases the calcium, now those messages get sent. So it determines the right time for sending those messages to the next cell. So now a little bit more about changes in the CACNA1A gene and these differences that can lead to different disorders. So a variant is just a change in a gene. A variant is the term that we tend to like to use over something like mutation because not all variants are harmful. But it's really just like a typo or a spelling change. So just some sort of difference. And again, most gene changes are completely harmless, but some can cause problems. And those that do cause problems tend to disrupt or change that gene's protein product. So in this case, it would affect the calcium channel itself. Missing or abnormal CACNA1A protein changes, the missing or abnormal protein for the CACNA1A changes the brain function itself. So you can see differences in how the brain works because that calcium channel does not work the same way. And then finally, changes in the brain lead to those features of the CACNA1A related disorders. So again, I have just sort of a little analogy here. So if we have a change in our recipe for the CACNA1A, this can lead to changes in our protein, which is kind of like the cake you're trying to make from your recipe. And those changes can then lead to changes in your bake sales. And that tends to be the thing that you see, those final features, and it starts all the way back up with changes in that recipe or changes in that gene. So then a little bit more about the different types of changes we can see. So one is called a missense variant. This is where one of those amino acid components gets swapped out for another one. So you can see at the top, there's sort of that original sequence. We can have our sets of 3 that change into those different amino acids that build the protein. And in the missense change, one letter is different. So instead of a U, you have an A. And that leads to one change in an amino acid from TYR, which stands for tyrosine, to ASN, which stands for asparagine. So just one component that gets changed. And this is the way that it can look on a lab report. So you have that P dot, which means that we're talking about the amino acid sequence itself. The TYR is our reference of what we expected to see. The position 130 means that's the number amino acid that we were looking at. So at position 130, we're expecting to see the tyrosine. And then finally, we see the difference. So we see an asparagine instead. So it's just showing that instead of tyrosine, we have asparagine at position 130. Another type of change we can see is something called a nonsense variant. So this is one where there's not necessarily a change in an amino acid, but there's an early stop, that it stops continuing to add new amino acids earlier than expected. So in this case, you can see it's still just one letter change that could have happened. So instead of an A, in this case, it's a G, but it puts a stop, and then it doesn't continue adding any of the other amino acids. So it's much shorter than it should be. And this is how this gets written. Again, we have our P dot, our TYR in 130, showing that we expected to see tyrosine at position 130. But then they denote that there's a early stop, either by a star or sometimes you'll see an X. So this is saying that that protein gets cut off now at position 130. Slightly more complicated type of change is a frame shift variant. This is one where the reading frame of those 3 letters at a time gets shifted. So often there's a letter that's added or a letter that's missing, and then it skews that whole set of reading 3 at a time. So in this example, you can see that the U is missing. So then instead of reading it UAU, GGC, CUA, it's now reading as AUG, GCC, UAU. So you can see that it actually can lead to many different amino acids being different because that whole reading frame gets skewed from that point forward. It can also change the stopping point for that protein. In this case, it's now going longer than it should because it didn't pick up that stop signal. A lot of times this happens where the stop signal is now earlier because of this frame shift change. So this can lead to many different amino acids being different, as well as a protein that's not the same length as it should be. And this is how this one gets written on the report. So first we talk about the first change that we see. In this case, that we see a methionine at position 130 instead of the tyrosine. That's what the MAT stands for. But then we note that there's a frame shift by writing the FS. And then we do the star 11 to show when the stop happens now. So in this case, it's saying 11 more amino acids later, there's a stop. So again, this can be a star or an X. But what this also tells us is that overall there's 11 amino acids that are different, and then a stop at a different position than typical. So a slightly more complicated type of variant is a splice site variant. And this relies on the way that different pieces of the DNA get stuck together to make the final protein. So there's like certain sections of the DNA that are used to make the protein and certain sections that are not, and they get cut out before the protein is made. So you can see in this case, the black lines in between the colored boxes are not included in that final protein, and then also this green section is not included. And the way that the body knows which sections to take is because it looks for really particular letter sequences along the way, and it knows this is where we cut and this is where we attach. So a splice site variant is like a change right at one of these borders where one letter becomes different, and then the body isn't sure which, or doesn't know which ones to include anymore. So it might include things that it's not supposed to, it might leave out things that it was supposed to include. So this could lead to changes where maybe you are now missing that yellow component, and you have the green one included instead. So the way that this gets written out is a little bit different. The amino acid sequence itself may not be different. So we use the C dot only. We don't have a P dot for this. And they're referencing the position in the final protein where this change was, and then it goes forwards or backwards, because often this is in a section that gets cut out. So in this case, it's saying position 235 minus 1. So this is saying it's the 235th letter that's included in the final protein, and then you go backwards one into an area that is not included in the final protein, and that's where our specific letter change is. So you can see this is sort of right at that border on the green one. It's into an area that's not normally included. And then we just showed G to A, showing what the specific letter change was. One thing I just want to note is that some of these splicing differences are normal. There are splicing changes that can cause problems, but there are different versions of CACNA1A in the body that in all people that we can have many different versions of the protein. So this is kind of showing an example. This is called alternative splicing, where all three of these might be perfectly normal copies of CACNA1A to have in the body. Some include certain sections and some don't include those. Certain ones might be more relevant to the body, and you'll hear a little bit more about that later on. But one thing that can be tricky is that when labs are trying to report what variant a person has, they have to pick one of these things to reference back to. So when they're trying to report what they saw, they have to pick one as their guide. So that can lead to some differences in the report based on which version they picked. So I kind of show here, based on how long a particular version is, you might have up to 36 letters, or maybe only 24 letters, or maybe 33 letters in total. And then same for amino acids, you could have 12 total amino acids, you could have 8, or you could have 11. And you can see, in this case, the orange circles are present in all three of these. They're in all of the copies, but in one case they're called number 11 and 12. In another case, they're the 7th and 8th amino acids. In another case, they're 10 and 11. So if you're referencing something that's with this first orange circle, it's present in all of these, but it might get a different number. So that's how you can have things like this example at the bottom, where the valine 1393 methionine is the same as the valine 1396 methionine. That's just because the reference that the lab was using to number where the change was a little bit different. So this is different than splice site changes. This is not anything to do with a harmful change or not, but it just can be a difference in how we reference things, but sort of related to that idea of the splicing. So our final type of genetic variant that we can see are deletions and duplications. So this means that there's an extra or missing piece of DNA. Sometimes these can be changes within CACNA1A where it's just one part of the gene is missing or there's an extra piece of it. Sometimes it can be the whole gene is either missing or there's an extra copy. And sometimes what can happen is that it's actually a much larger change, and so it might include the CACNA1A gene and then also several other genes. And those other genes might have an impact by themselves, like in addition to the CACNA1A gene. So again, these are just one other type of changes we can see kind of based on those changes within the chromosome itself. So now a little bit about how these changes can move in families. So CACNA1A disorders and the genetic changes that go with them can be inherited from a parent, or they can be de novo, which means it's brand new in a child. It's not been seen in any other family member before. This usually means that there was a change in one egg cell or one sperm cell that became that new child. The severe or early onset CACNA1A disorders are more likely to be de novo. They're less likely to be inherited through families. And again, some pictures of this. So you can see here, you only need one change in the CACNA1A gene to cause the disorder. That's called an autosomal dominant inheritance pattern. So if one person has a genetic change that actually is harmful, they will have the disorder. And then everyone gets one copy of the gene from their mom and one copy from their dad. So in this case, we have an affected person that has one typical copy of the gene and one copy with a change. And then whether or not their children have the disorder is a 50-50 chance. It just, like a flip of a coin, it just depends on whether they pass on the version that does not have any changes or they pass on the one that does have the change. So it's not, it's like a flip of a coin with each pregnancy. So it's not a particular pattern that you'll see in children. You can always have something like getting 4 heads in a row and flipping a coin. But on average, it would be about 50% chance or 50% of children with that disorder. When something is de novo, it means, again, brand new in that child. It's not something that we see in either parent. If we were to test the parents, we would not see it. It's something that happened in the egg or sperm cell that became that child. So it's not something that was really present in the parent, but it is present throughout that child. And in these cases, we don't expect any increased risk to siblings or more distant family members, but that child with the disorder, when they go to have children, would now have that 50-50 chance of passing it on. So now that they have it, they can pass it on. One situation that is a little bit different, especially when we're seeing something that's de novo, is something called parental mosaicism. And that's when a parent has some cells with the CACNA1A change or whatever genetic change, and some cells that do not have it. So this is something that occurs when, while in an embryo, the parent develops a change, and so then parts of their body develop with the change, parts develop without the change. So sometimes you might test a parent and you might see really low levels of that change. It's not suggesting that it's throughout their whole body, but it's suggesting that there's a small amount. That would be mosaicism. Another situation that can happen, though, is that maybe this genetic change is not present in their blood or it's not present in their spit. And so when we test them, we might not see it. But if it affects like their ovaries or their testes, they might be at risk for having more than one child that has this genetic change, because that genetic change is present in them, but just in those like egg or sperm cells that can be passed on. And once that egg or sperm cell has that change and it develops into a child, that child will have it throughout their whole body, because that original copy of DNA they received from the parents had that change. And so then it gets used throughout the entire body. So that's one situation that can happen, where even though a variant might look like it's de novo, you can have more than one child with it. And this is a rare situation overall. Usually when we see something that's de novo, we say the chance of having another child with the condition is less than 1%, but this is the reason why we can never say 0%. All right, so now a little bit more about the effects that these variants can have. So I apologize, the slides are slightly skewed here, but normal is when we might have both of the copies of the CACNA1A gene, the one from mom, the one from dad, both work as they typically should. And so you have your channels that are allowing the ions to pass as appropriate, and you have 100% of the normal amount of that. Loss of function is where you lose about half of that. So you only have about 50% of the normal functional activity of your CACNA1A channel. So you might be just missing one whole copy from a parent, or it's just not working at all. Gain of function is when the change causes a new property to be added to the calcium channel. So in this example, it's showing that more calcium passes through than it's supposed to. So in this case, the protein still gets made, but it's just not working the way that it was expected. So when we talk about the different variants, for loss of function, most nonsense, frame shift, and splice site variants are loss of function. Some missense variants are loss of function, and typically whole gene deletions are loss of function. For gain of function, they're almost always missense variants. Not all missense variants are gain of function, but most gain of function variants are missense. But rarely there can be other types of variants that could be gain of function. And we talk about the effect of this change on the channel itself. For loss of function, the effect overall is similar, that just not enough channels are working as they're supposed to. So you're missing some of those channels. For gain of function, the effects can vary wildly, and you'll see some examples from the functional studies that have been done about exactly all the effects that these can have. But some examples include that the channel might be too fast or too slow to open, or too fast or too slow to close. It might allow too much in, it might not allow enough in. Lots of different effects here. So this can in turn change to a different set of features that we can see in those individuals, or a much wider picture or more variable picture. So clinical features overall for having these harmful changes in the the CACNA1A gene could be learning differences, autism spectrum disorder, seizures and epilepsy, hypotonia, which is low muscle tone, ataxia, which is sort of a uncoordination of movements. You can have eye movement disorders, and you can have the hemiplegic migraine, which is simply, essentially like a temporary paralysis on one side of the body. For gain of function, these are more likely to have the seizures, severe epilepsy, and status epilepticus. Overall, it may be more likely to have the hemiplegic migraine and episodic ataxia, which is sort of these periods of uncoordinated movement. So these sets of features are ones that we typically associate with gain of function only, and we don't expect to see in the loss of function. But many of the other features can be present in both. So next up, we'll have Liz talk about how we can tell if a variant is harmful or not, and what the lab does on its end before reporting back on what changes they saw. So I just want to do one final acknowledgement of my team, including Olivia, who's here today. This is the CHOP Epilepsy Neurogenetics team, multidisciplinary team that's taught me lots about neurology and about CACNA1A. So thank you.
Good morning, everybody. As Lena said, my name is Liz, and I am a genetic counselor at GeneDx, which is a commercial diagnostic laboratory that does a lot of genetic testing for rare disease and also some common diseases. And we have identified a lot of variants in CACNA1A, mostly through our epilepsy panels, our autism panels, or our exome or genome sequencing tests. So I'm going to talk a little bit about when we find those variants, how we go about classifying them, and a little bit about the kind of follow-up steps that can be taken in certain situations. Just as an acknowledgement, I am an employee of GeneDx, and we were recently acquired by another diagnostic laboratory called Sema4 and are in the process of integrating with them. So I'm going to start with an overview, go through some of the evidence that we use to derive the classification, and then talk a little bit about some of the CACNA1A data that we have from the variants that we've seen at GeneDx, and then also talk a little bit about variants of uncertain significance and how we go about trying to resolve those. So our role as a diagnostic laboratory is to take the sample that's provided from the patient and the family. This is usually a blood sample or either a spit sample or a buccal swab, like a cheek swab. And we extract the DNA and we run testing. And when we're doing our testing, we're looking at the DNA. So we're not looking at the protein itself, which is the final product that's used by the body. That's technically much more difficult and would sometimes even require an invasive procedure to be able to do something like that. So that's typically only available in a research setting. Diagnostic laboratories look at DNA, which is something that we can extract more easily from other types of samples from the body, like blood or buccal swabs that are easier to collect. And so part of what we need to do is we sequence the DNA, we identify a lot of variants, and as Lena explained, some of them are going to potentially be harmful for the body, but many of them are not. They're benign. And so what we're trying to do with classification is figure out the difference, and we're using a lot of different information that we compile to try to do that. Laboratories all use a pretty consistent standard that's based on this initial publication from 2015 from the American College of Medical Genetics and the the Association for Molecular Pathology. And these two groups came together and said, hey, we're really starting to do a lot more testing. Before it used to be just for single genes. There wasn't great technology. Now we're doing a lot of really big gene panels and exome sequencing where we're looking at all of the genes that code for proteins in the whole body. We're finding a lot more variants, and we're seeing that there's a lot of inconsistency from one lab to another. So we really need to come together and come up with some standards. And so this 2015 guideline was the start of that. But the reality is that our bodies are complicated, which means that variant classification is complicated too. And so this simple framework has been an ongoing source of collaborative efforts to really try to refine these criteria and standardize the process, and that still goes on today. Most clinical diagnostic laboratories have either an automated or a semi-automated process for variant classification. So I'm going to talk about sort of the basis for how we do this, but understand that a lot of what I'm talking about is actually done now using computer algorithms and models and pulling in data and running it through bioinformatic pipelines that can collect all of this data and use the rules that are in the guidelines to help us come to a classification. Really, the purpose of all of this is to figure out if this variant in this gene could explain this patient's features, and that's ultimately what we want to put into the report. So there are 5 ways that we classify variants. These are the different categories. This stems from that 2015 publication, and so the categories that we use are pathogenic. These are variants that are expected to cause disease. Likely pathogenic, these are variants that are going to cause disease in nearly all cases, but we don't have quite as much evidence. Maybe we're seeing it for the first time. We don't have replication over time. So we're not quite ready to go the whole way to pathogenic, but we're really pretty certain that this is the answer for the features that we're seeing in the patient. There are variants of uncertain significance, which you'll often see abbreviated VUS, and that's what I'll call it through the talk. You'll sometimes also hear people say VUS or VUS, so if you've heard of either of those terms, we cannot agree in genetics about how we want to call this abbreviation, but they all mean the same thing. There are variants that are likely benign, and then variants that are benign. And so I present this in these buckets, but the reality is that this actually exists along a spectrum, right? And so when we identify variants, we, at first look, if we know nothing else about that variant, it's considered a VUS until proven otherwise. And what we're doing as a clinical lab is accumulating evidence to see if we can push that variant either towards the benign spectrum or if we can push it towards the pathogenic spectrum. And so that evidence builds over time and is what helps us determine the ultimate classification of that variant. You can see here at the bottom that if you think about this statistically, in order to classify a variant as being pathogenic, we want to have a greater than 99% probability that that variant will be pathogenic forever moving forward, no matter what else we find out about that variant. So we're very, very confident that this is the answer. Just to illustrate sort of the difference between pathogenic and likely pathogenic, the probability that a variant is pathogenic, if we classify it as likely pathogenic, is about 95 to 99%. So you can see that there's we're pretty confident that we have the answer, but we're not getting close to that 100% certainty that we have if we've called it pathogenic. With that said, because we have such a high threshold to get to likely pathogenic in general, most of the time if a variant is reported as likely pathogenic, a doctor will use that and act on it and treat as though that's the answer for the patient while we continue to try to accumulate additional evidence. You can see that that spectrum for variants of uncertain significance is very broad. And so we have these VUSs that fall sort of closer to that likely pathogenic spectrum, and sometimes we'll call those hot VUSs or suspicious VUSs. And then we have some variants that fall closer to the likely benign spectrum, where we're pretty certain that those variants will be benign. We just don't have enough evidence yet to get them there. And those you might sometimes hear referred to as like a cold VUS. So there's not a lot of evidence, and it probably at some point may be able to be reclassified. So there's a lot of evidence that can go into determining a classification, and I'm not going to go into detail about all of this. It's way beyond the scope of this talk. I could spend all day talking about this. But one of the most important factors that we look at, and the first thing that we consider, is the variant type. And so Lena talked about the nonsense, frame shift, missense, and splice variants, and that's incredibly important in determining the final classification. We also look at data from the general population, and this is also really important. So as you all know, CACNA1A disorders are rare. If we identify a variant, and it's very common in the population, say like 2% of people in the population have that variant, we know it can't be disease-causing, right? Because 2% of people aren't walking around with CACNA1A disorders. So knowing whether a variant is common or rare in healthy people in the general population is also very helpful information. We look at functional studies if they're available, and so we can't do these ourselves in the lab, but we rely on the researchers, and you'll hear probably more about some of the functional studies for CACNA1A here. And so we evaluate publications for functional studies for variants. We look at any other published data. So have there been other patients with this particular variant that were published in a paper in the literature, for example, that we could use some of that data to help with classification. We look at in silico predictors for missense and splice site variants. So these are algorithms that help us predict the likelihood that a variant might be pathogenic or benign based on the effect it's expected to have on the protein. We look at external databases, in particular one called ClinVar. So ClinVar is a database that is available through an NIH-funded project called ClinGen. And the ClinVar database is publicly available, and clinical laboratories like GeneDx and others send the variants that we've identified and the evidence to support our classification to these databases. So we may come across a variant that we have never seen before at GeneDx, but has been identified at a different clinical laboratory and is in ClinGen already with a classification and their evidence, and we can use that for our classification too. So this is a way that we collaborate as clinical laboratories to share information to help get better classification information for patients. We also can look at internal resources, so our database at GeneDx, to see if we've ever seen this variant previously. And then we also take into consideration the information about the clinical features for the patient. So we often are sent records or a list of features, and we try to make sure that, if we see a variant in CACNA1A, does the patient's features match CACNA1A? Because if it doesn't, then that doesn't really add up, and maybe that variant is not pathogenic and not the explanation. So each of those pieces of evidence is weighted. There is evidence that is supporting a pathogenic classification, and there's evidence that's supporting a benign classification. And so we take each of those pieces and we assign it a strength or a weight. So for pathogenic, we have very strong, moderate, and supporting. And then for benign, we have standalone benign evidence. So this is frequency in the healthy general population that I mentioned. If a variant is so common in the healthy general population, that's enough in and of itself for a rare disease to allow us to classify that variant as benign. And then there are strong and supporting levels of evidence that we consider. And so we take each of those pieces and we assign a category to it. Sometimes there may be evidence that starts with sort of a supporting category, but over time, we might be able to actually bump up to a stronger strength of evidence. So an example might be for those CACNA1A disorders that can be inherited, you could see segregation in a single family. So there might be a couple of affected family members that all have that variant, and that would allow us to say, okay, we're seeing this segregation in the family, we're going to assign some supporting criteria for that, because all of the people that have this disorder also have this pathogenic, or also have this variant, and all of the healthy people in the family don't have the variant. But it's only one family, and there's a couple people, and then later we identify that variant again in a second family, and there's a few more people, and we see the same pattern. We see a couple of people in the family with the phenotype have that variant, and a couple of people in the family that are tested that don't have it don't. And so now we've got two families, that evidence is stronger, and instead of using a supporting criteria, we could actually bump up and use moderate criteria supporting pathogenicity. And if we then found a third family, we could add additional evidence and maybe even get to strong. So over time, sometimes classifications can change because we started with a limited amount of information in that first family, but as time went by and we saw that variant again in more families, we were able to accumulate more evidence. So that variant might have been considered a variant of uncertain significance at the time we saw it in the first family, but by the time we got to the third family, we've got strong evidence towards pathogenicity. That might be enough to allow us to reclassify that variant and now call it likely pathogenic or pathogenic. And so even if someone has a report with a variant of uncertain significance, over time, we sometimes can change the classification and be able to classify those variants as likely pathogenic, pathogenic, or likely benign, benign. So we've assigned a level of strength to each of those criteria for each piece of evidence. And then we have this, kind of in the original publication, there was this guide that said, okay, well, now you've got all of this evidence and you've assigned it a weight. How do you actually decide then what the final classification is? And so you add all of those up together. So say you have a very strong pathogenic evidence and then you have some supporting evidence, that might be enough to get you to pathogenic. And you can use this table as a guide, and if it doesn't meet criteria to be one of those other categories, then it stays at uncertain significance. So here's an example of that. This would be a straightforward example where we see a variant. Maybe this is like a nonsense variant, and as Lena said, those are predicted to generally cause loss of function. The protein is short, it's very abnormal, it's not going to be able to work properly in the body. And so just knowing that we have a nonsense variant is usually enough for us to use a very strong pathogenic criteria. And then maybe we also know that that variant was de novo in that patient, so neither of the parents had it, which also supports its pathogenic. And then maybe our in silico models, so our predictors, predict it's pathogenic, or we know it's rare in the population, and so now we've got these levels of evidence. We have very strong, moderate, and supporting. And so when we add those up, you see that a very strong in combination with a moderate and one supporting is enough for a classification of pathogenic. So that variant would be classified as pathogenic based on the combination of all of that evidence. Here's an example where this is something that happens commonly with missense substitutions that we've never seen before, where we have very limited evidence. So for this variant, maybe we know there's a little bit of frequency in the population. It's not high enough for us to really apply strong benign criteria, but it's enough that we could apply supporting benign, because most variants in CACNA1A are not seen in healthy people at all, or are seen very rarely. And then maybe we've got an in silico model that's saying, oh, it's pathogenic. So we have two pieces of evidence, one suggesting pathogenic, one suggesting benign, but they're both only supporting level. It's not very strong evidence. And so this variant would be classified as a variant of uncertain significance because we just don't have enough information to go on to make a different classification. But over time, if we accumulated additional evidence and we were able to maybe add some benign, strong criteria and some additional supporting criteria, you see that that variant has now tipped. And really, the evidence that's supporting benign significantly outweighs the evidence that's supporting the variant to be pathogenic. And so we'd be able to reclassify that variant to likely benign. And so this is how we can sort of use all of that information together to derive that classification. So what about CACNA1A? So I looked at some information in our database at GeneDx about just in general, what kind of variants do we see? And so you see that 44% of the variants that are in our database are missense substitutions, so almost half, right? We see a lot of missense substitutions. And remember that those are harder for us to classify because we don't know what the impact is going to be on the protein and whether that would affect the way that the protein works or not. But we also see frame shift, we see in-frame, we see intronic. Those are both different variant types that can cause a splice site abnormality, like Lena had mentioned. We see nonsense substitutions. We see substitutions in the regulatory area, which is like out in front of the gene itself that has an impact on how it works. And so we see this broad spectrum. If you look at the variants that are nonsense and frame shift, so for us, those are the ones that are generally the easiest to classify because we expect that they're going to cause loss of protein function. About 7% of the variants we identify in CACNA1A are predicted to be loss of function. The other 93% we don't know, and so we have a lot of work that we have to do to try to figure that out. So then we can look at the classification of the variants that are in our database and see how have we classified these. So overall, about 8% of the variants in our database are classified as pathogenic, and another 6% are classified as likely pathogenic. So that means that about 14% of the variants, now remember these are variants, not patients. So some of these variants may have been seen in a lot of different people, right? But these are looking at the variants themselves. 14% of the variants have been pathogenic or likely pathogenic. 32% are classified as likely benign, and 16% are benign. So nearly half of them are in that benign spectrum. And the remaining 37% are of uncertain significance. And so those are the ones that we still know as a community we have more work to do to try to figure out. So you can actually look at the classification spectrum by variant type. So remember that the nonsense and frame shift variants are predicted to have a big impact on protein function. So they almost always are classified as pathogenic or likely pathogenic just because of the type of variant that they are and the effect that they're expected to have on the protein. We do have about 3% of frame shift variants are of uncertain significance. And usually those are sort of out near the end of the protein, or they cause a very subtle change, and maybe it's not significant enough that we are able to say for sure it would cause loss of protein function. So there are those instances where a nonsense or frame shift variant might still be of uncertain significance, but that's clearly much less common. And most of the time with those variant types, we do get an answer for the family in the report. It's very different with missense substitutions. So about 3/4 of the missense substitutions in our database are variants of uncertain significance. About 13% are pathogenic or likely pathogenic, and 12% are classified as benign or likely benign. And then if you look at in-frame substitutions, so these are substitutions where there's a few nucleotides, a few of those DNA pieces are missing from the protein, and so maybe one of the amino acids or a few of the amino acids are missing, but it doesn't totally disrupt that protein. Those also can be challenging to classify, similar to missense variants, and about half of those are variants of uncertain significance. If you look at intronic variants, actually most of those are benign. That's because the only intronic variants that matter very much are the ones that are very close to the exon intron that impact splicing and are going to impact the way that that protein is made. So the the 6% of variants in the introns that are pathogenic or likely pathogenic occur in those positions that are right by the exon-intron boundary, but we actually sequence a little bit farther than that into the intron, and so we have variants in our database that we've identified in that intron that we know are benign, and so we have a lot of benign intronic variants. It just kind of gives you an idea of how important the variant type actually is for classification and how we're much more likely to have a VUS result, especially with a missense substitution, whereas we're much more likely to be at path or LPath with nonsense or frame shift. So if we've identified a variant of uncertain significance or we don't have a definitive answer, what can we do? So if the parents, the biological parents, were not tested already, usually that's our first recommendation. So let's try to figure out whether that variant is inherited or de novo, and that often is enough to give us the information that we need to be able to reclassify a variant. If this is a situation where there are multiple affected family members, instead of just testing the parents, often we'll offer to test the affected family members and see if we're finding that variant in all of the similarly affected family members or not, and that segregation information can be important. Sometimes we have no clinical information as a lab, or maybe a baby is very young and we don't know sort of what that clinical picture is going to look like over time. So sometimes as there are new or changing clinical features, that can help us with variant reclassification. Sometimes it's that we see the variant in a different family, so we're getting some additional information from that family and we're able to combine that information together. Sometimes it's functional analysis. So this is not something that's done in our laboratory, but done by researchers who you're going to hear from here, who publish their data and share that information. And we can use that. There are some criteria that actually look at functional analysis, and that is often very important for us in classifying missense substitutions. For variants that we think might impact protein splicing, but we're not sure, we can do something called RNA studies. We do this on a limited basis in our lab, and it's also done in research labs, so sometimes that's an option, only for specific variant types, but certainly can be important. Sometimes it's a better understanding over time of a gene and how it influences disease, and so it's not something that changes with the evidence we have, but with how we apply it, because we understand more about that protein and how it impacts disease. And sometimes it's that down the line we see that variant in other patients and we know that there's other variants. So if, for example, we identify a CACNA1A missense substitution in a patient that has an SCN1A pathogenic variant, we know that patient has an SCN1A pathogenic variant, and that's going to explain many of the features. That means it's much less likely, not impossible, but certainly much less likely that that variant in CACNA1A is pathogenic, and we could apply some supporting criteria towards it being benign, and sometimes that's also helpful with classification. So with all of that, I just wanted to sort of end with a quick summary of the kinds of information that can be included in laboratory reports. And so generally, there should be a summary of clinical information from the literature or from our internal data, if that's available. If there's published functional data, including a brief summary of the functional data, and with all of these references to the actual publications for anyone who wants to review in more detail. We provide information about the predicted effect that we think the variant would have on the protein, and then a summary of any of the other data used in classification, so the population data, anything else like that that would have been used. Things we get asked about that sometimes we can't provide as a laboratory. So Lena mentioned that missense variants can be gain of function, loss of function, or mixed. So that's not something that we can tell from the sequencing that we do or from looking at the DNA. That can only come from functional data. And so if there's published functional data, we can include a summary of that, but we can't make that prediction just knowing the variant itself. We generally can't provide long-term outcome data or prognosis, so we can give a summary of what we know in the patients that have been identified so far, but that doesn't necessarily predict what's to come. And we can't provide information about effective treatments for a specific patient because we're just not to the point where we have that information. And moving forward, probably that gain of function, loss of function, mixed impact is going to be important for all of that. And so those kinds of things you can't get from a lab report. It's really looking for trying to make the diagnosis and say whether this is actually the explanation for the features or not. And that's kind of the limit to what we as a diagnostic lab can do. And why all of the other speakers here are going to be so important, because they're taking that information and they're taking it to the next steps and pulling all of that together. So with that, I'll end. And I think we have a few minutes, so Lena and I can take any questions. Thank you.
Virtual participants who are entering questions in the chat. We don't have a whole lot of time, so we'll do one virtual, then one in person, and sort of back and forth until we run out of time, which is not going to take long. Here is our first virtual question. We've now seen papers that talk about patients with mutations that are both loss of function, meaning there's a decrease in the amount of channels, plus gain of function, increase in the sensitivity of the channels to differences. So it seems like perhaps it's not as simple as just gain of function or loss of function. Is that something that you can comment on?
Yeah, that's definitely true. There can be mixed effects. I didn't talk about that in my slides, but there definitely can be mixed effects where there's some aspects that match loss of function, some aspects that match gain of function. Again, when we're kind of looking at a channel, there's many different ways that you can measure it. I think some of the later talks will get into that a little bit more about what these mixed effects can look like. But certainly, it's not as simple as just loss and gain of function. So again, it can be really complicated when trying to fully understand especially what some of these mismatch changes actually do to the channel.
All right, it is 8:58, so I think we'll have time for one question. Oh, okay. All right, I'll go over to Dr. Jen over here.
Thank you for a very clear presentation. I have a quick question for Liz. What does the 14% with pathogenic and likely pathogenic diagnoses translate to roughly, the number of patients?
That is a good question. I don't have the raw numbers, and I don't want to guess and provide you with the wrong information. I do think that Sunitha and Lisa have also been collaborating with so we have a team at GeneDx that works with our patient and family support groups to provide information like this upon request. So I think that there is ongoing discussion with them and Amanda Singleton, who's one of our genetic counselors with that team, who has and can provide that data. But I don't know it offhand, so I don't want to misspeak.
Thank you so much.
Yeah, thank you.
All right, we'll go back to a virtual now.
We have someone who's asked if their genetic testing was done years ago, and at the time it said a variant of unknown significance. How do they go about getting it reclassified or finding out if it has been reclassified, short of redoing genetic testing?
Yeah, that's a great question. So actually, my suggestion would be to have the provider that ordered the testing, or if they're followed by genetics and they're working with a genetic counselor, reach out to the laboratory that did the testing and ask them if they can reevaluate that variant. And so we do this all the time at GeneDx. We actually go back and we'll do an assessment with the information that's currently available to see if anything has changed and provide an updated report if there's a new classification. There also are some things that families with a VUS might want to think about doing. So another one is through the NIH-funded ClinGen project. There is, for example, something called Genome Connect, and so you can register there. And if clinical labs submit their information like we do to the ClinVar database, if they note that there's been a classification change for that variant, they will actually also proactively reach out to the patient and let them know. So that's another option that families might want to consider as a way to get updates. We've definitely had some families that have gotten an updated classification and report through that process too.
I think just to also to add to what Liz said, and Liz, you can correct me if I'm wrong, but there's some degree of this that can be automatic, that if a new patient presents to the lab, has the same genetic change, and the lab is able to update the classification at that time, then they'll automatically send out new reports to all patients that have had that genetic change before. But that doesn't, that only matters, it only comes up if there's another patient that has that same change where they're looking at it again, or something happens and they're looking at it. So it's always great to reach out to the clinician, to the lab, to ask them to specifically look at it again, just in case the lab hasn't already done that.
Yeah, and I would say every 1 to 2 years is probably a good frequency for that kind of a question to come, because these things do change over time. And that usually gives us enough time to acquire more evidence.
So for the final question?
Okay. On that note, what kind of coordination is there between GeneDx and other labs? So not all of our families get their testing done through GeneDx, obviously. And is it just through ClinVar? Like you rely on each of the labs to submit that information to ClinVar, or is there some other process?
That's generally the best way. So all of the larger diagnostic labs that do testing for disorders where CACNA1A would be included on panels do submit information to ClinVar. So that actually works very well for rare disorders, because the labs have all agreed to partner and collaborate in that way. So that's the primary way. Occasionally we will have, there's a complicated variant, a tricky variant. I mentioned that sometimes we're sort of on the border between uncertain significance and pathogenic or likely pathogenic, and we know that that could make a big difference for management. We sometimes have proactively reached out to ask another lab if they've seen a variant and had data. I know we also get questions sometimes from clinical genetic counselors who have a report from a different lab, and they will contact us and ask if we've ever seen the variant, and if so, how we have it classified. And so sometimes we get connected to another laboratory through a clinical genetic counselor or other healthcare provider who's reached out to ask us about a variant that was found in another lab initially.
I will say on ClinVar, some labs report more information than others. So for, I think both GeneDx and Invitae are ones I've seen a lot, where they really do give all the information that they had. Some other smaller labs might not give as much information, but it can be really helpful that there's a lot of evidence that they'll include in those submissions.
Liz, you mentioned during your presentation that when you're looking at variant classification, you look at prevalence in the general population as one of the factors. Mm-hmm. When you're doing that, are you looking at the general population primarily in the US or in European data sets, or are you looking globally? A lot of our families have, they're not recurrent variants, and so they want to know, is there anyone out there that has the same variant as my child or myself? Yeah. And I'm just wondering how accurate those databases are from a global perspective.
Yeah, that's a great question. So the primary resource that we use, well, there's two primary resources that we use for this. One is the publicly available NOMAD data set, which is pulled together from a combination of many different exome and genome sequencing studies. It is currently heavily skewed towards people with a European white background, and is not as well represented for people who have other backgrounds. And so the data that is in NOMAD currently is definitely better and more helpful for people who are from those white populations. Really what that means is we're more likely to have data in NOMAD that could help us reclassify something as benign or likely benign, whereas for people from other ethnic backgrounds, it's not in the data set. That doesn't mean it's pathogenic. It just means we don't really have a lot of data. And so it might be a common variant in people from a particular country or a particular region of the world that's just not in those data sets. There are a lot of efforts that are happening currently across the genomics research spectrum to try to pull together larger studies that include more of a representative population from around the world. And so I think over time that data will be available. And that's also part of, I think, as those data sets get better, how we'll be able to reclassify variants because we'll have more of that information. We also use our internal database, which, so when we have done exome or genome sequencing and we request parent or relative samples to look at in comparison to the patient, if those individuals are healthy, we have that information in our database as well. And so we do look to see if those variants are common in our data set. We mostly test patients from North America, but we do test patients who have samples coming from some places in the Middle East. And so we do have, I think, probably internally a better data set that's more reflective of the Middle Eastern population than what's available currently in NOMAD. That's an excellent question, and definitely, I think, a place where we need more information moving forward, just to help in general with classification and other efforts.
Thank you, Liz. Does anybody else have a question here? All right. Well, let's thank Liz and Lena one more time, and then we'll move on to our next speaker.